don't think about this as a science project. Never start a company thinking it's a science project. It's a very different thing for an academic versus a biotech business person. It's not a science project. What is the end goal? How much capital does it take? Is it going to provide value to patients? Number one, is there a product to be had? Number two, if I build that product, is there a buyer for it? Number three, what's the fastest path to heaven to get there?
This is the Nucleate Podcast, the voice of the next generation of biotech leaders.
Welcome to the Nucleate Podcast. My name is Sam Kessel, and on this episode I speak with Raj Devraj, venture partner at Atlas Venture and CEO of Rectify Pharmaceuticals. Previously, he co-founded and was chief scientific officer of Disarm Therapeutics until its sale to Eli Lilly in 2020, and was CSO of Padlock Therapeutics until its sale to BMS in 2016. Before that, Raj was president and chief executive officer of Pharmaceuticals and senior vice president of Rectify Pharmaceuticals. Prior to that, he was senior vice president of Jubilant Drug Discovery and Development. Raj spent 14 years in positions of increasing responsibility with Pfizer Global R&D and legacy pharmacia and cereal companies. He has led multiple discovery and early clinical development teams that have advanced 8 candidates into clinical trials for refractory cancers, rheumatoid arthritis, diabetic nephropathy, COPD, and pain. Raj received his BS in pharmacy from the University of Mumbai and PhD in medicinal chemistry from Duquesne University. We had a great conversation on this episode, and we hope you really enjoy it. Raj, thank you so much for joining the Nucleate Podcast today.
Yeah, thank you, Sam. I appreciate the opportunity to talk about drug hunting, the venture capital system, and also a lot about Rectify and what we're building at Rectify.
Fantastic. So let's start at the beginning. You originally grew up in India, and you went to the University of Mumbai where you studied pharmacy. Can you speak to what it was like growing up there, studying there, and how you ultimately transitioned to doing your PhD in Pittsburgh?
Yeah, I think India is a land of hustlers. You got to really stand out. I think it took me a little bit long to sort of find my feet. I did find my feet. I was pretty good in academics. Things came very easily for me once I started to apply myself. And it's a land of hustle, right? You need to catch that bus, think about New York, except with 10x more people. So from a very early age, you thought about being, quite aggressive regarding where you need to be, your goals, and how do you hustle and get the attention that you need to continue to advance yourself or anything that you're passionate about.
That's great. And you can see that throughout your career that you did hustle your way through, you worked hard and grinded your way to the top. So ultimately, after you transitioned to your PhD, you also pivoted to industry when you came to the States. So you spent some time at cereal companies and Pfizer for a decade. Curious what inspired that move to industry, and if you had any advice for a current PhD trainee that is considering making that leap as well.
Yeah, I think now I'm dating myself, but we're talking. My PhD was a very multidisciplinary PhD. It's chemical biology, what it's called now, was more what I did in my PhD. So I did a lot of synthesis, a lot of drug design, a lot of cell-based assays, really learned a lot of therapeutics and pharmacokinetics and pharmacodynamics and pharmacology and action of therapeutics. So got a very broad sort of view. I never considered myself to be a specialist ever. And I never felt comfortable working in a very specialized science. Nobel Prizes are given to certain people because they are so indebted in terms of really trying to hone their craft and really work on a single problem for a very long time. That was not me. I fall in love with many different problems at the same time. So it was the best path for me through to the industry. It was not an easy time when I started to look out for jobs in the industry. It was not easy. But, I think my breadth of my knowledge, how I approached the interviews that I did with Pharma, and I got many offers, 3 different offers, and I chose Searle. Searle spoke to me. It was not a very large behemoth company. I was not looking for that. At the same time, I knew at that time biotech scene was not at all emerging. So it was, where do I work in a smaller mid-sized organization with a very dynamic R&D leader who will bring people along with him. And that was Phil Needleman. And we'll talk a little bit more about Phil Needleman, who's been my mentor for a very long time. So, so that's what I joined. And for new PhDs, thinking about a career, I know a lot of PhDs who go through now do a very long postdoc, and it's pretty very long, especially for biology and biological sciences. A little less for chemistry and chemistry-related sciences, pretty long for chemical biology as well. So it is very important to sort of align your goals and set realistic expectations at this point in time. A, this is going to be a slog. B, you need to be committed to putting in a lot of work and not getting paid a lot in return. But you're doing this because you're really passionate about science, you're passionate about the research thesis that you're working on, and you're able to contribute in a very novel and creative way to advancing the science that you're working on, whatever the science you're working on. I was always drawn to drugs, pharmacy degree, so it was very easy for me to get drawn to drugs and making new drugs, and that's what is my passion all my life. I would say to students, be patient, be realistic. It takes longer, and it's been like that in the COVID and the post-COVID era. After the brouhaha in 21 and 22, after that things have settled down dramatically, and with the current economic macroeconomic environment, it's still going to be pretty slow until the industry finds a footing for itself, the XPI finds a footing for itself, and things all move back up.
And I think that advice for patients does also apply to your experience in entrepreneurship as well. It is not a fast process. You mentioned the XBI. It seems like a difficult time right now in 2025. Recent tariff policy obviously has had an impact in the past few weeks here. We're recording this in April of 2025, so tariffs are biting. Curious on what you think of the challenges of navigating a more risk-averse capital market right now. Venture seems to be risk-averse. Public markets seem to be risk-averse. How would you think an entrepreneur can navigate this environment?
Yeah, I think, listen, we are all facing that. Making the dollar go a longer way is the best way right now you can work. I think, to me, platform companies are very, very difficult to fund. Um, there are unicorns. Let's not forget about Zara and the other companies. There are unicorns for sure. But I think in totality, I would say if you have a really strong product thesis, you have very strong target validation, and you have a strong team, you stretch that dollar to go as long as you can. And that has been, I have multiple different experiences building different companies, and I can step on that in a little bit as well when we come to that section. It is not easy, right? Right now, the markets, I think the public markets have been really badly hammered over the past year and continue to do so now, right? The XBI is really down in the dumpers. But I would say even the private capital markets, right? Capital flows down. Finally, it gets into the private capital markets as well. And the private capital, venture capital is also few and far in between. And things are going to take much longer to get funded. If you're able to fund your company through very discrete seed goals, do those. Work a lot, build your equity in the company as a founding team, maybe 3, 4 founding members. Build it. Take it as much as you can through the seed phase. That means you can do 300 experiments. You got to choose what are the 3 to 4 most important experiments we can do, how long is it going to take, what is the titrated capital I need, what does the business plan look together, and at the end of a year, in the end of 6 months, at the end of 9 months, at the end of a year and a half, what is that set of data that is truly translational that allows me to say that I have a drug product out there. And I'm purely talking about drug therapeutics right now. I'm not talking about reagents, tools, and med devices, so which might have a different thesis. So.
That makes sense. So being ruthlessly capital efficient, operating with leaner teams than you may otherwise in a better funding environment, and prioritizing the experiments that you need to do, which ones are truly the most critical versus which ones are nice to have, not need to have, is even more important when there's a long fundraising cycle and it's more challenging to raise.
Yeah, it is easy to say, do the killer experiment. Guess what? With teams, it's very, very hard to execute it and make it a reality because everyone has a slightly different bent on it. And then it really depends on the detail, right? Do you trust your chief scientific officer, your chief medical officer, your translational person to really understand, is that data truly game changing for the company to move forward to the next step of capital raising? Good ideas will get funded. Good ideas get a lot of diligence done. I mean, we are at Atlas, I know we are stepping through a lot of great ideas and great companies and figuring out how we can capitalize them to the next stage of milestones.
That makes a lot of sense. So I want to talk about Atlas now too. So you spent a decade at Pfizer and started out at cereal companies like we were talking about, and then you spent some time at Decipher.
Yeah.
Decipher. Thank you. And you mentioned your mentor, Dr. Needleman. So curious, how did your path unfold after Pfizer, getting into earlier stage biotechs, and ultimately, how did you get to Atlas?
Yeah, let's take a step back chronologically. I would say I have a 30-year career in the industry. And I would say if you put me in a small bucket and give me a one-liner, I would say I'm good at translating breakthrough science to products. That's what I do well. And then, of course, over a period of time, I've also understand the business of science and how do you capitalize companies and what sort of milestones you get and what is the negotiation you have with investors to help deliver the milestones required for them to get the financial exit that they require, right? So 15 years in large pharma, Searle became Pharmacia became Pfizer. So I was part of one company. It was just 15 years in that. And then the last 15 years has been building biotechs. I think I needed to move from large pharma to a biotech company. But truth be told, I was very concerned about moving to a startup company because I didn't know how the environment of a small biotech would be. So for me to go from the behemoth of Pfizer, where I had a very large group, to a company which had 4 to 5 people was a little scary for me. So I went into the next best step, where you had a company of 50 people or so, which is a biotech company, well funded, and try to go there and learn what it takes to do business development. What is the meaning of portfolio analysis, portfolio strategy for Decipher? Decipher, as you know, has become a very successful company with 2 marketed products and has been bought out by Ono. A long cycle of innovation there, like many other companies that have been bought out. And that taught me a lot. And when it was time for me to move on at that time, I was keeping in touch with Phil, and Phil said, Raj, I like Atlas. Phil was very close to Bruce Booth. They both served in the same scientific advisory board for UCB, and said, why don't you go talk to them? They'll probably need a person like you who's, adept at drug hunting. I'd worked in a lot of projects in Pharmacia, Searle, and Pfizer that went all the way through to phase 2 POC. So very good from idea all the way to phase 2 POC, and how do you build teams and analyze the data through the clinic. It was a very long conversation, right? You need to get comfortable. The investment team and the partners need to be comfortable that they are making a bet on an entrepreneurial person who still has the needed expertise and experience to take the companies to the next step. I joined Atlas as an EIR. And there I met Michael Gilman, who was the CEO of Padlock Therapeutics. Michael Gladstone, Michael Gilman, and myself, we started Padlock together. Mike was a fantastic mentor as well, a superb storyteller. Learned a lot from him on are how you're able to tell the story of starting a new company. What are you working on? What are the key things? Start with the big vision in mind, and then step through the details of the science and the milestones and the funding ask. So I learned a lot from on the ropes with Padlock. With Padlock, we were very fortunate. We were working on a class of enzymes called the PAD enzymes, protein arginine deaminase enzymes, that were in vogue, and we could advance a thesis of using these PAD4 selective inhibitors to help with rheumatoid arthritis. BMS liked what we had to put together, and they bought us up, right? And it was a preclinical exit. It was a really nice exit for everyone involved. Atlas founded it. Bruce was the chairman of the board for that, and others participated. So it was a great exit for us with BMS. A lot of other companies also had put interested in the company, and we chose BMS. After the sale of Padlock in March 2016-ish, I was going to take some time off. I spent time with another venture capital firm. I won't say what. Spent some time, but I was drawn back to sort of the high science of Atlas, right? I was not interested in companies that were building very large platform companies and firms. I was more interested in the therapeutic thesis. So while I was thinking about it, I was still in EAR at Atlas, attending all these meetings. At the same time, Phil called me back again, Phil Needleman. So here's sort of a process that I've used all the time. I've always been hired back by people who I've worked with. And that's how I landed in Decipher as well. The chairman of the board I worked for at Pfizer. He was the head of R&D at Pfizer. And that's how I landed at Decipher. Then Needleman called me back and he said, Raj, why don't you talk to two MD, PhD neurologists at WashU? They're working on this class of enzymes, SOM1, which they're really pioneers on the target validation and biology. And I was like, Phil, I'm not a neuroscience guy at all. I'm like onc, immunology, inflammation, pain. I can handle those. Neuroscience is a completely different beast. He said, no, just come out and talk. And Phil is not going to suffer fools. So when Phil says something, I go and talk. So I did a lot of diligence myself, and I bought the thesis of building a company that can work on SOM1 inhibitors back to Atlas. We seeded the company, raised a $30 million Series A, and I was sort of the acting executive there, right? Lead executive for the company for a while. I founded it, became the CSO for the company, and we could exit again Disarm and sold that to Lilly in October 2020. Again, preclinical, we had some nice data stepping through. And SOM1 is a very, very good target that really is an important target that addresses axonal degeneration. So you can truly have a disease-modifying therapy for both peripheral nervous system indications as well as central nervous system indications, ALS, for example, right? Where axonopathies are a key sort of cause of morbidity and mortality. In January 21, I became a full-time venture partner at Atlas Venture. During the strategic transaction we were undertaking for Disarm with Lilly, I met with the founder of Rectify. Again, I was asked to meet with Jonathan Moore because I was connected, my senior VP and head of translational sciences at Rectify and him were close friends. Jonathan Moore had started his own lab at MIT after working 28 years at Vertex. And he wanted to really understand how we could extend this entire platform of small molecule drugs that can be correctors or potentiators, right, beyond CF gene, beyond this cystic fibrosis gene, which is ABCC7. So I was intrigued. I talked to him. It was again Atlas, and we put a diligence team together. What was most important for us is what are the first targets we work on. And how do we take the biology risk early off the table, the target biology risk? And I'll come to those themes that I've always used in my career. Is how soon can you take the biology risk on the table? How soon can you take the modality risk of the table? How soon can you take the translation and then therapeutic risk of the table? And what are those key experiments or data sets that are available that you can step through to really convince yourself that there is an investable thesis here? So we started on a few programs at Rectify. With Rectify, I was a board member of Rectify. I was not the CEO. And then I was also board member of a couple other companies. And then by September 21, we could raise a very large $100 million Series A led by Atlas, Omega, Forbion, and Longwood came in. And we started building the company out, right? This is a different company than what I was used to. Disarm and Padlock and Indelo Therapeutics, where I was on the board, was more of a very titrated target, small molecule, advancing towards a translational data set and then to the clinic. With Rectify, we had to build out a platform. And the platform we built out is this positive functional modulators, which are small molecules that are able to restore or enhance membrane protein function. Not the most easiest thing to do. Let's just be very clear, right? Science is never going to be easy. And you needed to build a platform. So we needed to step through three things. Number one, what are the assays we'll use? High throughput screening, and what are the mechanistic assays? Because we were only interested in advancing those PFMs that directly bound to the protein. We were not interested in finding chaperones or mRNA modifiers, transcriptional regulators. So we needed to build that assays and mechanistic assays. Our library started out small. Now it is a very privileged small molecule library that is highly enabled and shows 40 to 60 x increased hit rate for membrane proteins. And then, of course, we had a cryo-EM platform enabled by the founder of Rectify, Jonathan Moore, who was a structural biologist. And we have less than 3 angstrom structures of every membrane protein that we have worked on, both APO as well as ligand bond. So the platform started. It takes a lot of investment in the platform. And truth be told, it got very expensive. Operational costs became quite expensive post-COVID. Things are not easy, right? Even small sundry items like turning the lights on, janitorial services, water services, including doing CRO costs at China, all started to move up because of supply chain disruptions post-COVID. And we had to bear the brunt of that, right? So we needed to start to think about very quickly, is there a path forward for a product? And that's my interest. And my interest is not building out large platforms. So very quickly we pivoted, and our lead program is a product for multiple hepatobiliary diseases. And our lead indication is for a very large unmet hepatobiliary disease, primary sclerosing cholangitis, which is about 60,000 odd patients now, right? I wouldn't be surprised if that prevalence continues to go up, much like what PBC has done, right? Once you have a good therapeutic, there'll be a lot more patients who will be found with PSC as well. I'm absolutely convinced of that. And our target is a dual targeted PFM for BSEP and ABCB4. So these are two targets. BSEP is ABCB11. So basically, it's a very selective positive functional modulator that enhances BSEP and B4 wild type function, but also can correct the mutations of BSEP and PFIC and B4. The reason we chose those targets initially was BSEP is the etiologic driver of a rare hepatobiliary disease, pediatric, called progressive familial intrahepatic cholestasis 2. B4 was similarly an etiologic driver of PFIC3. So very strong human genetics that allowed us to say, okay, if we hone in, can we move a product forward? And we have superb data with our dual targeted PFM showing we can correct the mutations and really completely modify the disease in our models. But as you know, PFIC2 and PFIC3 are very small indications, right? Small, rare, monogenic indications. Of course, they can sustain a very high price point, but the IBAT inhibitors from Mirum and Albrio, which is now at Ipsen, were doing well. So how do we compete with something that's so far out, 5 to 6 years ahead and in the marketplace, and it's doing pretty well? They're not disease modifying, but they do very well in reducing pruritus and fatigue that are the main sort of phenotypic effects that's seen with PFIC2 patients. These kids, they're terrible. They continue to itch, and it's very, very, very bad. And the IBATs work really well for those. So we moved, and that was a pivot for us as well with this company. I would say moving from PFIC2 as the lead indication, and then understanding that our PFMs actually can enhance wild type function, gave us the strength to go back and interrogate the larger rare hepatobiliary diseases, which are PBC and PSC. And our compounds work beautifully in PBC and PSC as well. So our lead program is now at IND enabling stage, and we are moving into the clinic very soon for PSC.
Fantastic. And there's a lot to unpack there. I want to go back a little bit
Sure.
to Padlock and Disarm, which you talked about briefly. And I want to talk about how that connects to what you're doing today at Rectify. So I guess my question for both of those. So both Padlock and Disarm had successful exits relatively early on in their lives. So I'm curious on, was that the goal all along? Did you do something specific in terms of management to position for an exit? Because I assume there's a bit of a difference for building a company for an exit versus building a company to be standalone. So I'm curious on how you made those key decisions. And then let's connect that to what you're doing now at Rectify too.
For sure. Happy to. I know I spoke a lot. I was very passionate about also talking to you about the new company. So yeah, let's step back. I think you need to think with a slightly different lens on how big the company will be that you're building. Number one. Number two, what is the first set of funds that you got into the company, sort of the Series A fund, and what is the initial goal of that Series A? What were you planning to deliver? And then be very open to surprises in the data. That is the key, right? Being prepared to be at the doorstep of innovation and taking advantage of that, and continuing to do BD, continuing to talk about the science, continuing to make sure that the science and the innovation and the data sets are presented in the right manner, with the right pitch at the right audiences. And those are the things that a large firm like Atlas allows for entrepreneurs, right? They're very well connected to the industry, along with entrepreneurs who might be connected, like myself now, right? When I started, I was not, right? I knew, of course, everyone in large pharma knew me, but then investors didn't know me, right? So how do you sort of build that legacy of experience and expertise so you can continue to open doors for your new companies? With Padlock and Disarm, we didn't build large teams. We had a very small group of very seasoned drug hunters, program managers, business professionals, finance professionals. And we knew is there a goal to do a strategic exit early? But you wouldn't know that until you've had 3 or 4 conversations and you had the first set of data. With Padlock, it was quite fast. Within about 18 months, we had a good set of translational data that we could talk to people about. We were ready to finance the company. Listen, you don't build a company thinking you want to exit it early. Never do that, right? Never do that. But you titrate the capital enough, and you don't hire a very large team unless you're working on a large platform, which we'll talk about at Rectify, right? So we outsourced most of the science at Padlock and Disarm to external CROs. So what became important for us to hire was people who could manage these CROs, drive biology, drive chemistry, drive translational modeling. What are the CROs you can trust? How can you sit on the calls? How can you step through the data? Is this the data of good quality? If not, how do you retool? What is the statistical significance you need to have in those studies? So it's a completely different set of people you're looking for. You're looking for senior managers and senior scientists who are able to seamlessly do that work with CROs. Rectify was different. We needed to build a platform, and we needed to really build the assays out internally. These are not easy assays to do. It took us a while to hone in our high throughput screening assay, which is a hybrid screen looking at protein expression. And then we had to build a set of junior scientists who could come in, work in the lab, generate the assay data. And that was very, very important for Rectify. So Rectify's sort of founding thesis was to build a platform and then work on multiple products. And we have worked on multiple products. With Disarm and Padlock, it was a very titrated target. Inhibitors of PAD4, PAD2, inhibitors of SAM1, and finding applications for those. How do you translate high science into new therapeutics? With Rectify, how do you de-risk the platform, then find the 3 or 4 pipeline programs that you can advance, and then ask the question. Rectify is a more expensive proposition. Disarm and Padlock are less expensive, but that's normal. It's nothing here or there. It's just reality of the situation and how you build companies and how you think about raising capital to build those companies, right? So
Yeah, and that's very interesting. The different approaches for more asset-centric companies, which you had with Padlock and Disarm versus a platform. And I think a lot of our listeners in the audience who are in the nucleate activator program are often faced with this decision, which is going back to the topic we started at earlier, in a capital restricted, situation where it's harder to get investments. How do you build your company in a way, whether you're pursuing one asset and trying to advance that, or continuing to invest a bit in your platform, generating multiple assets like you are at Rectify? It's a very interesting and important strategic decision. It sounds like, since you all are backed by Atlas, you have the capital and the ability to develop the platform and multiple assets. Still sounds like it's pretty capital efficient as far as it goes, but it's a different approach than what you were doing at Padlock and Disarm, where every dollar goes into advancing a single asset.
absolutely 100% correct. Yes. But listen, I think at the end of the day, investors are looking for products. Products are what, number one, products are important. We work as entrepreneurs, as drug hunters, to make sure that we have new therapeutics to really help patients, number one. Number two, products are what generate interest with pharma, right? Having a good product and stepping through the early clinical data is what generates true interest, right? Yeah, you do get lucky because, and I would say lucky is the wrong word too. You're very sort of deterministic about the data sets you will collect, and you're always having early conversations with people, right? Most of my time is spent having conversations with pharma analysts, bankers. I'm talking to them almost on a weekly basis. So that's another thing that I would give to entrepreneurs, is that if you don't have that network, find a firm that can provide you that network so that you can start to build those relationships early, because analysts see a lot of things. They'll provide tremendous advice. Bankers see a lot of things in the markets. They provide a lot of advice. Pharma, you have to continue to talk to them, seeing what you're building is of interest, right? And that's what we got to do. Sometimes it's very high science. With Rectify, there are programs that we're building that are really, really incredibly novel ways to treat diseases, right? Disease modifying therapeutics for multiple hepatobiliary diseases. Next program is to reduce vascular calcification and prevent renal insufficiency in CKD. Completely new approach, right? So these are all things that take time to incubate in the minds of pharma, find the right champions, both science-wise and business-wise, to get deals done.
And it also connects to what you were talking about earlier, with your mentor being important, with your relationships being important. It sounds like the people you've connected to, whether Dr. Needleman or the pharma connections you brought to bear, have been extremely helpful as an entrepreneur, in that you can't operate alone. You can't just be like the classic imagery of a entrepreneur in a garage doing it themselves. It sounds like you've had a lot of help along the way. You've gotten mentorship guidance, you've gotten feedback from different groups that's maybe they're not telling you what to do per se, but it's informing what you do, and it's giving you real-time information to make the best possible decisions.
200% correct. I think it takes a village, and no one's successful. I mean, It's crazy. They say success has many fathers and mothers. Yes, it does. Uh, on the flip side, they say failure is but it does too, right? This is an industry that we got to face the odds of failure is very high. Successful products are very low. So how do you sort of make your bets early on? And if you choose to continue to invest in a program through to IND, to me, the most important question I ask myself is, what is the fastest path to clinical data? And I'm extremely interested in understanding, is there a proof of mechanism data set I can get? Proof of concept, yes, everyone talks about proof of concept, but there are two important decision points before proof of concept, right? Proof of biology, proof of mechanism. Slightly different things, could be same for certain targets, certain mechanisms, but slightly different. And to me, I'm really hone in on what does the proof of mechanism look like? Can I get that? Can I be very creative in the clinic and get it with a very small check? Very small amount of time, very small check. And then ask that question before I step into the larger 3 month, 6 month POC trials that might be required to really understand whether the therapeutic is truly meaningful benefit in that desired patient population.
And I also want to talk a bit about your indication selection. So, you already mentioned the novel platform with the
Sure.
positive functional modulators you're using at Rectify, and you talked about how it's being used first in hepatobiliary diseases.
Right.
What I find really interesting is that, based on my experience at Locust Walk, we see a lot of interest in CNS diseases or your more classic autoimmune diseases like rheumatoid arthritis or lupus. I haven't seen a lot of hepatobiliary companies recently, and I think it's a really interesting and counterintuitive bet to make that you're pursuing that. So I'm curious, I'm assuming it's based on the science, but if there was also, when you're choosing multiple options for how to take your platform, why primary sclerosing cholangitis and the other hepatobiliary diseases you're looking at, why did those rise to the top in terms of how you're using your platform?
Simple answer first, science first. It was really driven by target validation and human genetics. And then also knowing if we, beyond human genetics, because PSC and PBC are large, rare, common diseases. These are not genetically driven diseases, but the step through of the two important targets we're working on, BSEP and B4, and the mechanistic, the function they provide in A, BSEP effluxing bile, if we modify that positively, and B4, effluxing phospholipids that help coat the bile and make them less toxic to the biliary epithelium. We could pull on both those livers, and the functional data set was very critical for us to advance. So we were not in a position as a company to say, let me work on autoimmune disease, let me work on obesity targets, let me work on muscle sparing target, let me work on Alzheimer, PD, ALS, and force fit a target into that. We needed to build out the platform, and we organically followed that. But truth be told, I think it would have been fun actually to go back and revisit that. We were just marching down, we had 3 programs that we were investing in, and all the 3 programs have made tremendous advance. I mean, you talk about CNS, we have a program in CNS, ABCD1, D2 for XALD and AMN, a very large rare neuro opportunity with large unmet need. As you know, there are some gene therapy products that have a lot of issues and black box warnings around cancer and other things. So we are very excited about those. But if I go back now and if I had more capital, I would choose to work on an obesity target, a brand new obesity target that does one thing. If I positively modify that, I know I have a benefit and differentiated benefit, right? Of course, it's a completely different thesis. I got to figure out what translational data I need to generate to tell me, based on the onslaught of the 65th double G, triple G company, or the new amylin company, or the GLP amylin company, or the activin receptor company, how do I differentiate myself? That's number one. Number two, I would choose CNS targets. I would definitely choose two targets that we're very interested in, but which are large investments required to go through the clinic for Alzheimer's, ABCA1 and ABCA7. If I positively modify that, I think I have a very good shot at starting to address the mechanistic thesis of ApoE lipidation and its importance in clearance of A beta. So I think I would have done that. So these are sort of decisions you make, but predominantly our decision was based on target validation, building the scientific platform, and organically flowing our lead product and target product profile to those targets and indications that are supported by that target. Yeah.
That makes a lot of sense that you follow the science first. And for what it's worth, those are both, the lead indications you're looking at, hepatobiliary diseases. There are significant unmet needs there, right? There is an argument for why your drug is going to be used. So obviously it is a clear rationale. Maybe it's not going with the crowd of where everyone else is going to your point, into what I was talking about in terms of market trends. But I guess at the end of the day, as long as you're following the science and there is a meaningful unmet need to address, does it really matter where the rest of the market goes, as long as you're able to effectively develop something that makes patients' lives better in a meaningful way?
Oh yeah, I keep telling my team all the time to be three things: be resilient, be courageous, and be extremely innovative, right? You can't spend more money than you can't spend money you don't have. It's an expensive proposition, building a platform company, choosing indications, working on the targets, working on small molecules, stepping through safety. None of that is easy. But in the onslaught of the news of every week that there's another company that has been bought out for $1 billion in phase 1 obesity data, weight loss of 20%, weight loss of 8%, weight loss of 15%, and then of course differentiation thesis as well, and then tremendous amount of innovation coming from China and other places. You chose an indication. You're very strong and passionate about it. You have a good plan through the clinic. Stick to it. Good things will happen.
I love that. So when you're thinking about the trajectory of Rectify and what you're doing, and sticking to your guns, as you're saying, where do you see the company moving in 5 years? It sounds like you alluded to there's certainly potential new options you can use the platform for. It sounds like your focus will be on your 3 primary assets, but curious on where you see the company in the next few years.
Right. So yeah, we want to do partnerships. Okay. So the number one goal now that the science is matured and we have been talking to people is to understand if there's some partnerships to be had to bring. I'll never say anything is non-dilutive, right? Because when you give out an asset early, that is dilutive to the asset, but you bring in some capital to help advance the most important thing for you, which is your lead program. So in 5 years, I think Rectify the data, the POM and the phase 2 POC data that we're going to get in PSC patients. And I hired a very seasoned and experienced rare drug disease developer, Paul Boudet, CMO, who really came to the company because he was interested in developing this drug for hepatobiliary diseases. He was the CMO for Cimabe and helped develop Seladelpar for PBC. So he knows the space well. He's connected to the KOLs. It was very good for us to attract him to the company. And he's very interested in taking this company and getting a quick POC study done. I think there's a tremendous I think push to get that POC data is going to be very transformational for the company. Either it's going to be transactional for the company, or it allows us to get to the IPO market. I think we can things have to open up. I know today no one is interested in talking about it, but markets change, right? I can always read the Twitter feeds or LinkedIn feeds of those who are always talking about the biotech markets in a swoon. Sure, at this point of time, 2-3 years ago, it was on a high, and that cycle of doom and gloom will go into the high up. So things are going to change. I think we just need to be capital efficient and continue to be extremely focused in delivering the lead program, and then decide strategically, what is the other program we want to fund? Or is there another way to do the transaction for the company? Because our pipeline is an incredibly valuable pipeline. That pipeline can be used to find positive functional modulators for ADPKD, positive functional modulators for an obesity target, MCR4. All these things are there, right? So how do you monetize the platform? What is the best corporate infrastructure to monetize the platform if it's not for BD? These are things that we have a conversation about all the time. But current focus, get the lead program moving into the clinic. Let's collect data. Let's see what we can do with the other two assets that we have in there.
Fantastic. And I'm curious, since you're mentioning, business development partnerships, something I spent a lot of time with at Locust Walk, what do you think makes a successful partnership at the end of the day when you have an innovative technology partnering with a larger company that you have experience on both sides of, right? With your main significant amount of time at Pfizer, and now significant experience in the biotech world. How do you think both sides can benefit, and how can both sides make a drug succeed ultimately?
Right. So we stepped through immunology, we stepped through obesity, and I would add oncology there and maybe some CNS diseases, that are hot. Those are easy to find a very receptive audience because that's where the money is. You're going to have FOMO. No large pharma cannot participate in the obesity market, right? Everyone's going to look for an asset and take part in the tremendous sort of economic cycle that the anti-obesity drugs are going to bring to the industry. But when you go to other disease areas, it takes longer. What you have to reach out to is reach out to the hearts and minds of the scientists in the organization. You need to be sure that you have a champion in an organization and build that very early and lead them and be persistent and almost on a quarterly basis, if not on a twice a year, provide them a sense on your progress. And that really helps sell your new target, your new sort of modality. Although it's small molecule, it's a new modality, right? You got to start to talk about why that mechanism is important, why the target is validated, and how it can cure the disease. Number one. Number two, you got to find the right industry. Who are the right industrial partners who are interested in that segment of the disease? And really hone in. It takes a very long time. It takes a personal touch. It takes traveling. It takes shaking hands and actually presenting in front, just not on Zoom. I think those are all the things that are very important. So go to JPM. I know everyone talks about JPM, but for entrepreneurs and for biotech companies, it's a great way to get a personal audience in front of people. And the more senior they are, the better it is for the biotech company. So I come back to the first word I use, hustle. BD is not only a science, but it's also an art, especially in doing it in new targets, new platforms, new therapeutic areas. Takes a long time to find a scientific champion and then bringing along a business development champion internally who can put a deal together for you.
Absolutely. So building those deep in-person relationships is critically important. I know Zoom is very common nowadays and Teams or whatever video platform you choose, but sounds like that in-person and really informal relationship that you have with people, the personalities matter. Getting to know each other matters. Obviously, the business part matters too, the science. Does the deal make sense for both sides? But making sure you're in front of the right people on a regular basis, making sure they're up to date on your data, that personality, that relationship allows the partnership to potentially emerge over time. And it sounds like it doesn't happen all at once. It's not an overnight success. It's something that builds over time that as a startup, you might be too early for a pharma partnership, but if you build a real relationship and the science plays out well, there's a real possibility of that partnership to emerge, assuming you've done the legwork all along the way to smooth that out, it sounds like.
Right. So just to harken back, when I Disarm exit was October 2020. The real detailed conversations with Lilly that I sort of engineered in started in August of 2019. Started that, and we went to JPM. JPM, we had a great discussion with the senior leaders at Lilly and January 2020, and then the world shut down right after that. And then after that, it was all done through Zoom, but took more than a year, 15 months of building the relationship, talking to them about the target, and helping them sort of drive that diligence process. It takes a long time, long time.
And I'd liken it to a marriage in a way that it's a long-term relationship in BD. In some ways more complicated than M&A because you have two separate entities who may have different motives in some ways, work together on a shared goal. So finding that common ground can be challenging when you have different needs at a small company than a large company. I'd assume different cultures, although with your experience, I assume you're able to bridge those cultural gaps fairly well.
Try to, right? Try to. Listen, what a biotech might call a DC might be very different than what a pharma calls a DC. But what a biotech calls an IND is pretty surely the same as what a pharma calls an IND. So people have to realize that, entrepreneurs and especially biotech entrepreneurs, when you say a DC is a DC, what do you really mean? What's the data set that you have? What is the translational data sets that you have? What is your accurate prediction of human dose? And what is the safety margins you have, right? As we all know, there is no drug that is super safe. It's all about the dose. And the drug is all about the dose. So how do you talk about the safety margins? How do you proceed forward? How much strength and conviction you have that you got your human dose projection right? What's your starting dose in the clinic? And how can you step through the efficacious dose range? These are all very important questions to ask.
So I'm curious. So for our audience not familiar, DC is development candidate.
Correct.
What do you think the major difference is between the way a small biotech thinks about that versus large pharma? I'm guessing the smaller biotech might have a looser definition. It might need a little more iteration before it gets to the IND stage. And I'm assuming a pharma is going to have a lot more requirements in terms of data readouts before calling something a development candidate.
No, that is true. I agree with that. So I remember back in Pfizer, we had two classifications. DC needed to have 1 week, minimum 1 week safety studies in a rodent and a non-rodent species, right? Biotechs might not do that. Some biotechs, capital constrained environment, might call a DC after a 1 week in rodent and acute dose escalation MTD in a non-rodent, right? But if there is a case to be made in a certain therapeutic indication, or you have to step through the dose range, it might be better for you to collect that data. Typically, I don't see pharma holding out that you need 2 weeks, but I think now I'm hearing through the grapevine, through my BD work, pharma is also looking for 2 week data. That's a lot. That's a DRF study. That's a dose range finding study in rodent and non-rodent. That's a lot of capital for a biotech company before it is done. So it's a negotiation, right? Everything in the world is a negotiation. For CEOs, for CSOs, for CMOs, it's a negotiation internally with their investors, right? For private companies. It's a negotiation with the board of directors if it's a public company. And then with BD and pharma, it's a negotiation as to what you have done and what is acceptable for them, right? So is the next check of investment worth spending to really step through safety? Because you've got all the data in translational sciences, you have initial safety. I don't think it's worthwhile. I think you got to be street smart about this.
Now, this leads into an interesting topic around what are the cultural differences and what are the main differences, we'll call it, across big pharma and smaller biotechs. You've been in both for a decently long time. So I'm curious what the biggest differences are and what you feel like the major learnings that you had coming from big pharma and bringing some of those learnings to a small biotech that someone without that background would not have.
Scientific rigor in large pharma is absolutely true, because you got endless amount of capital, and you can continue to ask questions, right? Of course, all large pharma, we had aggressive goals to deliver DCs and INDs, right? And you got to figure out how to do it or make quick kills. But you can absorb quick kills and kills in pharma a lot more than in biotech. With biotech, you have it's a negotiation with investors. You have a titrated amount of capital to a certain milestone. And if you're not able to meet that milestone in the set time with the set amount of capital, it's a negotiation, right? And those are not the easiest conversations to have, but it's a very different. The scientific rigor and the amount of data you can collect for a new modality or a new or to advance a program into a new therapeutic area is a lot more compared to biotech. It just is. It's just purely got to do with two things: amount of capital, amount of time, and I would probably third one I forgot. I would say the team. Right? You have access to a lot more experts and domain experts in pharma that you have to go and seek out in biotech, right? Um, in biotech, I think the most important advantage we have in biotech is speed to decision making. I think it's so much easier to make decisions in biotech compared to pharma. In pharma, you got to go up to a value chain, up and down, yeah, and it takes time to schedule meetings with senior management, right? Um, and that is not going to be easy, right? There's a lot of preparatory conversations, preparatory decks going through, and it takes just much longer in large pharma. But you need to know, walking with your eyes in, that is going to take much longer for you to get a decision from pharma versus biotech, where we can all just turn around the round table and say, here's the data that came through the email. Let's talk about this data, what that means to our program and our lead program or our backup program, and how do we want to think about investing it. So speed to decision making, extremely, extremely efficient in biotech. Amount of capital required and the amount of data you can generate, much, much higher in large pharma.
And that makes sense for how the different styles of communication differ, timelines differ. Obviously, pharma is more like pivoting a cruise liner, whereas a biotech, you're looking on at a little motorboat. So it's a lot easier to move,
Exactly.
but it can be a little bit more challenging in terms of the resource constraints. So that's a really fascinating difference. And it kind of explains why the cultures are so different. In pharma, you want to make sure every box is checked. Whereas in biotech, you need to move fast because you have a limited amount of money and time to make your decisions.
Yeah, for sure. 100% agreed.
So when it came to starting Rectify, how did you vet this idea initially of the platform? How did you do some of those early experiments, if you did any prior to starting the company? How did you test the waters and figure out whether this was a good fit to dedicate substantial amount of your time and energy into a new company? Obviously, you're at Atlas, and there's probably a high bar to move forward with something. But I'm just curious, what was at the initial stages that gave you conviction around it? What kinds of things did you do prior to actually starting the company?
Yeah, I think let's speak about the scientific founder was Jonathan Moore, right? 28 years at Vertex, and was intricately involved in understanding the mechanistic details and the structural biology of the Vertex CF candidates. So that's exactly what we're doing. So it was ABCC7 is the protein product of the CFTR gene, right? So what he was trying to do is writing up grants to see whether he can extend that to other ABC targets. ABCA4, which is an etiologic driver of Stargardt. Everyone is working on Stargardt as a gene therapy approach. And we were thinking about starting a new program, small molecule, for Stargardt as well. And then what we did is, okay, when I started talking to him, he was setting up a high throughput screening assay and understanding, okay, if there's a high throughput screening assay, how reliable and quantitative it is to pick up compounds that show increased protein expression, number one. So what we did is, even before we put a seed in the company, a small seed check in the company, four of us spent a lot of time boiling the ocean on ABC transporters. What we found is there are 48 human ABC transporters. 21 out of the 48 have etiologic mutations that drive rare monogenic disease. Right there, you had a tremendous substrate to work from. Then you got to boil the ocean further. Okay, which are the ones that are valuable moving forward? Do any of these ABC transporters have a very long tail of mutations? Are some more concentrated? Are those mutations the most prevalent mutations that cause the disease? So we started stepping that. Number three, is there a market indication? Is there a translational model available? If we develop a drug, is it truly disease modifying? Because you're curing the disease, right? Because you're correcting the mutations that drive those diseases. These are all things that we did even before we put a seed check. Then we started putting the seed check, and then we started to ask the important question. If we put a library of small molecules together, can we A, develop assays for a few ABC transporters? Number two, if we take these small, very small set of libraries, can we actually find drug-like small molecules that increase protein function? A, potently, two, selectively, right? And that was sort of the first goal of the seed program. Once we got that, then we started to really advance the thesis further and further. And the next question became, okay, now we got these compounds. Sure. How do we know if these are just broad spectrum chaperones that we are not interested in, that are working on some sort of a chaperone pathway that is not going to be safe as we move forward in safety? And how do we know these are not working at the RNA level, but actually acting at the protein level? So then we had to de-risk the two assays, which are mechanistic assays, to ask those questions. So those were the first things. And then we started quickly building out the library based on prior history in the literature and prior history in IP. So boiling the ocean in the IP, knowing what are the correctors and potentiators out there, and then starting to pick off and building a large set of library, small molecule library compounds. So those were sort of the series of steps we went through. Redoing the high throughput screening assay, understanding whether we can find, if we develop it for 3 or 4 ABC transporters, can we find small molecule hits for those? Number three, are those small molecule hits worthwhile pursuing? Right? Not easy to do. It took us a while. It took us a little bit larger check than I would have liked, but that's where we were. Yeah.
And as you outlined earlier, the potential of this platform is pretty impressive and applies even broader than just rare monogenic diseases, as you alluded to, with both primary sclerosing cholangitis and primary biliary cirrhosis. Right. I'm curious, what made those come up in terms of the science? So how did you figure out, you said you were starting with rare monogenic diseases. How did you realize you were able to apply it to conditions that didn't have monogenic?
The first inkling, right? When we started the company, it was a founding thesis of rare monogenic disease. So we were very interested in screening variants, right? Mutational variants. So let's say we had a BSEP variant E297G. We'd screen that. We would find compounds that increase the protein expression of that. Then we'll step through our mechanistic screen. Then we'll say, okay, this is actually working at the protein level. Then we get a crystal structure. We confirm that the compound that we found is actually binding to the protein and changing the folding configuration of that protein as it gets synthesized from the ER. And then we had to show function. What we found out was very interesting. We found out that not only do our small molecules correct mutations, but when we screen them in primary human hepatocytes, we are able to increase wild type function of BSEP and B4. So that immediately opened our eyes. Oh my God, now I could take the walk from rare monogenic to large common diseases, right? I can address the mutations that cause rare monogenic disease, but I'm also able to increase the wild type allele if I have a haploinsufficient disease, or increase wild type function. Now, with the third thesis, though, it is just not important to get comfortable with the cellular data. You got to go in vivo. So we had to build the right translational model and ask the question in a normal sort of translation model. If I give my compound and I dose it orally, over a period of time. For us, it was 6 weeks or beyond. What are the number one? We had to find the disease model. The disease model shows a significant increase in liver enzymes and other pathology associated with PBC and PSC. And with a small molecule, can we address all those different things? And we saw very broad spectrum efficacy with the compound. But I tell you though, culturally, you need to be very courageous to do that experiment. That is an expensive experiment. Think about it. 2 doses, 6 weeks, control experiment. And that was what is required from people who were very experienced in the company. So the senior set of scientific leaders said, let's do that experiment. Because although the fundamental thesis was around rare monogenic disease, let's go and start asking the question. And that's what opened our eyes. The data that we got was phenomenal. And we continued. Now we have a backup molecule, we have a lead, we have a backup. And not only are we thinking that the backup might be an insurance policy for the lead, but we might be able to take it for two completely different indications. So just a different mindset. So that courage to take that decision at a very rapid manner and being at the cusp of innovation. As soon as that innovation occurred, we took the decision to start to ask that question in vivo.
And you had to have conviction on the mechanism and see beyond the immediate relevant data there in monogenic disease and say, hey, this has potentially much broader applicability. And while it may be expensive to run this experiment, it does open massive doors for the platform and worth doing earlier.
Right. And it was interesting for us because, when you said open the platform, that was the insight and data and innovation that helped us really advance the thesis that our small molecules are able to bind at a very productive region in these transmembrane proteins. And we could use the structural biology insights and do hotspot mapping. We could take those and say, okay, are these 3, 4, 5, 6, 7 different hotspots that we find working on ABC transporters transferable to other membrane proteins? And that's what led us to say ion channels, GPCRs, solute-like carrier proteins, and we found these hotspots. And then we started to take very small sets of library compounds from our own Rectify library and screen. And we found, wow, we can increase wild type and we can correct mutations of other membrane proteins. And then it's about just dialing in the selectivity, right? You need specificity for that target to the small molecule. So that really allowed us to expand the platform to new target spaces.
That's fantastic. Now, for someone in our audience who's, at the earlier stages that we're describing with Rectify, that there's a lot of big open-ended questions, right? There's so many different ways you can apply a technology or a platform. What's some advice that you'd have for one of the entrepreneurs in our audience that's considering making a big strategic bet, kind of like what you did, going after broader prevalent diseases, moving beyond monogenic diseases? How would you recommend someone approach that decision before taking action? Because sometimes if you have limited money, that bet you're making does have big consequences for your company. So what is your thought process? Like, what is your problem solving approach for making those decisions?
First thing is there a target product profile that you can get excited about? Start with the end in mind. What is the therapeutic area that is of interest to you? You have expertise in the therapeutic area, or you're a general R&D person, or you're a business person, doesn't matter who it is listening, right? Whether they're business, finance, is there a therapeutic area that you feel? And I would say, of course, it's easy to pick a therapeutic area, but you're not an investor, you're an entrepreneur. It's easy to pick an investment area where it's hot right now. But remember, innovation takes 3, 4, 2 years sometimes if you're lucky, but count on 3 to 4 years to really know whether you have a decent product before you can move it to the clinic. By that time, things are going to change. Okay, so have conviction in the therapeutic area, in the disease you're targeting. Build a TPP early. Understand, is there a target product profile? what is the modality that I need to pursue? If I work on this mechanism, is it really going to move the needle in the disease? Number two. And number three, if I have and meet a clinical POM or POP, is there a receptive audience that's going to transact that program? You're not an academic, you're not in large pharma, you're working for investors. You need to make sure that you also have that mindset. You wear that cap. Is there a transaction to be had, or is there a BD deal to be had? At the end of the day, investors put money in because what's most important is to get their returns back on investment, right? That's the key for them. So how do you ensure that you take that back? No, I'm not saying it's easy. And I'm not saying things, well, science takes, sometimes fails. Sometimes science is more expensive. And the business of science is sort of shaky. Capital markets come and change. Investors, patience will change. So having that gumption, making sure that you have that hat on as well. Number one, do you have a target product profile? Number two, do you think your mechanism is going to move the needle in the target? Number three, what is the modality you're going to use, right? Because we can talk about antibodies. Great. I think antibodies are an engineering problem. Small molecules are not. You don't know what a small molecule is going to do until you go through safety. To me, the translational data is important, but as important is safety data with a small molecule. It's so critical to make sure that you have enough margins to move forward for that indication, right? And then you need the experience and expertise. So those are the things I would consider as an entrepreneur working in biotech, trying to work on new therapeutics. Absolutely be killer. Surround yourself with the right people. If not, surround yourself with the right analysts who will provide that feedback to you. And one of the things with experience in 30 years in the business is I'm connected. So I can bring that experience to bear to help me understand what's going on or not, right? Analysts, bankers, pharma people, investors, they're all giving you feedback. Trust them. Get the free feedback. That's easy to get because you're going to put a lot of money into actually generating the data. But feedback and opinions and insights are free. Seek out, build relationships, and get them early. And step through like 3 fundamentally important questions you're going to ask about it. Don't think about this as a science project. Never start a company thinking it's a science project. It's a very different thing for an academic versus a biotech business person. It's not a science project. What is the end goal? How much capital does it take? Is it going to provide value to patients? Number one, is there a product to be had? Number two, if I build that product, is there a buyer for it? Number three, what's the fastest path to heaven to get there?
That's fantastic advice. So a lot of things that you covered there, keeping the end in mind, having a goal target product profile, making sure that you have good relationships with people who can provide you feedback on that vision and make sure that what you're doing is realistic and of interest.
Absolutely.
Any last words of advice for the entrepreneurs in our audience who are taking the leap to start a new company? You covered a lot on the scientific as well as the personal side, but just curious, any final words of advice for our audience who are considering going out there and starting their own company?
Get a planner. Get a strong, detailed planner. Look at the budget that are going to put through, trust them. Initially you have to be involved in driving the budget and driving what is that investment that you need to drive from point A to point B. Be realistic about the tranche sizes, right? Every investment is going to come tranched, right? That's how people hold you accountable. Sometimes you miss it, sometimes you don't. But in this business, very, very, very hard to predict. Don't shoot yourself in the foot if you cannot predict the right milestones, timelines required. Science has a way to keep everyone very humble. But so very important, get a planner. If you come from a scientific background, provide that planner the realistic understanding of how much it's going to cost, how many people you need, how long it'll take, to get to a milestone? What is the most important de-risking milestone for that company? What are you trying to remove as a risk? Is it biology risk first, or is it a known biology? And is it a modality risk? Is it a known biology and a known modality, but it's a differentiation risk? So what does that data look like in the preclinical world? And then as you step through, what are the safety studies you need? Plan that, have a budget for. So align yourself with a very, very strong business analysis, planning, operations person if you're not that person. If you're a strong business, finance, operations person, align yourself with a very, very strong scientist. Make sure that what you're saying is rooted in reality. Your reputation matters. And that is the key thing. You never want to lose your reputation in front of investors, pharma, anyone. It's science is about seeking the truth. So be true to yourself and have negotiations. It's always a negotiation with investors. It's always a negotiation with your team and your management team. And show tremendous trust. The first founding team, the 3, 5, 6 people who sit around the table and make those tough decisions and go out pounding the pavement to get money are the most important people you're going to be with for a long time. Consider them family. Treat them like family. Show respect. You'll get that respect back.
Fantastic. And on that note, Raj, thank you so much for joining the Nucleate podcast.
Thank you, Sam. Thanks for providing the opportunity to talk about a varied amount of topics. We took a long time, so look forward to it and continue to be successful at Nucleate, and look forward to other conversations as you seem fit.
Absolutely. Thank you.
Thanks, Sam. Bye.
Thanks for listening to the Nucleate podcast. Be sure to subscribe if you'd like to keep up with future episodes. See you next time.